My name is John Cullen, and I want to tell you a story. It's a story about a scandal, broken relationships, gossip, rumors, money, corporate rivalry, and curling. It's the story of Broomgate, how a single broom, yes, a broom, turned friends into foes and almost killed the 500-year-old sport of curling.
It was a year I'd like to forget.
Broomgate, available now.
Frequency Podcast Network. Stories that matter, podcasts that resonate.
If you ask anyone who has been touched by it, you will quickly understand why ALS is seen as such a horrible diagnosis. It's progressive, it's devastating, it takes everything from its victims, and it's incurable. For now. When you interview a lot of medical researchers, you get used to them shying away from the term breakthrough and being completely allergic to touting anything in the early stages of research as a cure for any condition. Which is why a Canadian-led discovery, one that nobody right now is shy about calling a breakthrough, is giving so much hope to so many ALS patients and their loved ones around the world. But beyond hope, this research offers us a unique look into a decades-long fight against one of the worst diseases humanity has ever confronted. And it gives us a glimpse inside a world of modern, cutting-edge research. So how exactly did the researchers make this breakthrough? How does the treatment work? How close are we to saying the word the world wants to hear? Cure. I'm Jordan Heath Rawlings. This is The Big Story. Dr. Michael Strong is a professor of neurology at Western University's Schulich School of Medicine and Dentistry, as well as the Arthur J. Hudson Chair in ALS Research, which is what we're discussing today. Hello, Dr. Strong.
Oh, thank you for having me.
You're most welcome. Why don't you just start for listeners who have been lucky enough not to have been touched by this disease? Give us the Coles notes on ALS. What is it? How does it affect people? Who does it affect? And how many Canadians have it?
So right now, there's something in the order of about 4,000 Canadians have this disorder. It is one of the big three of aging. So when you think about Alzheimer's disease, that's the one that comes right to the top of your mind, Parkinson's. ALS is in that same group. So this tends to be something that starts around your fifth or sixth decade of life. Although we're seeing it in patients now well up into their eighth and ninth decades as well, which is a bit of a change. And the big problem with it is that the nerve supply going out to the muscles, for whatever reason, dies off. And so the muscles pay the price by getting weak, wasting, they do some twitching. And ultimately, over the course of 3 to 5 years, that wasting of muscles also affects the diaphragm, and individuals will die from a respiratory process. So it's not a great one.
Obviously, the answer to this is just generally unpleasant and not great, but maybe you could describe what it's like to live with this disease or to care for an aging parent or loved one who's diagnosed with it.
So it's certainly for the individual themselves, it's horrendous disease in the sense that you're, in essence, ultimately locked in. By and large, most aspects of your cognition are preserved. There are things that we've found that suggest that there are some subtle differences that can occur over time. But for the individual, what they're experiencing is that they gradually get locked in. So that's probably one of the worst possible things that you can think of happening to somebody. But on top of that, then it also affects their speech and their ability to communicate. So they're unable to speak. Some of them start that way, some individuals, and what we call a bulbar onset. So that's in your speech and swallowing component. So imagine all of that, and then you're the loved one or the caregiver of looking after somebody. So, that really is the issue, is that, this is a disease that affects an individual, and in doing so affects the entirety of their circle, whether it be their children, their loved ones, their parents. And the age range, even though I spoke about it as something that's sort of later on in life, I've taken care of patients, as we all have, in their late teens, early 20s and 30s. So this can affect young people just as much in some senses.
A lot of people will know this disease as Lou Gehrig's disease. It's been diagnosable for decades now, I guess probably nearly 100 years. And all this time, what kind of treatments have we developed? Are there any treatments that currently exist?
Yeah, there's lots of things that we can do. And I like to break those things into, what can I do, I collectively as physicians and caregivers, to actually stop the disease versus what can we do to actually help you during the course of the disease. And it's in the latter half that there's been a lot of work done over the years, particularly the last several years. So for instance, things such as simply addressing respiratory challenges. So the ability of your diaphragm to compensate for your breathing when you go from a sitting to a lying position. You and I would presumably have no difficulty in doing that. The diaphragm adapts to that loss of gravity and such. When that diaphragm is starting to be paralyzed because of ALS, then you can feel extremely short of breath just lying down, and it disturbs your sleep patterns. So we use things, for instance, like what's called non-invasive positive pressure ventilation, or BiPAP for short, which is, very close to the same thing we use for treating snoring. But what we found is that, and again, when I say we, I really mean the collective researchers, worldwide on all of these things. If you do that early on in the course, when it's first starting to become a symptom complex, you can extend survival by almost a year. And you improve cognitive function better because people are getting a restorative sleep. We do the same thing for dietary supplementation. We know a lot about what to do in terms of physio and those types of activities and such. And there's a lot of medications we can use to try and help with the symptom complexes as they, like for instance, not being able to swallow salivans.
Right.
So on the one hand, it's been very clear that there's been a significant change just in the day-to-day management. And perhaps one of the biggest things on that is the establishment of specialty clinics, which sounds like, well, everybody's got a specialty clinic. But, there's been clear evidence to suggest that if you are a patient with ALS and you're followed by an experienced team, it also will extend survival. So these are all things that have been incremental improvements in the disease course, certainly even during my time as a practicing neurologist. Then you go to the second bucket, which is the treatments. And our track record on treatments for ALS has not been good. There's been a lot of really good science, and those drugs tend not to work. So we only now have a very small handful of drugs that seem to change the duration to some degree in some patients, but nothing that stops them.
This brings me to obviously why we're talking to you today. I don't know if you would call it a breakthrough. I've seen it referred to certainly as a breakthrough in headlines. Maybe just outline what kind of work you guys were doing and what you discovered.
So the work has been really since the inception. Certainly I started doing my research into all of this, was to try and understand the nature of why do these motor neurons die off. And one of the hallmarks when we look under the microscope at the spinal cord after somebody's passed away from this, and we see the remaining motor neurons there, within those cells, we see what looks like a train wreck of material sitting in there. They're like this big aggregates, or they're almost like balls of yarn all woven together and spread, and they don't belong there. And one of the major proteins that are part of that, and a protein is just the end result of what your DNA tells you it needs to make. One of these major proteins is seen accumulating like this in over 95, 97% of all ALS cases. So it is the single most common protein in there. And we know that when it starts to get caught into this train wreck, there's a lot of other things that are going on in the cell that are leading to its death. And we believe that the fact that this protein is where it is one of the major drivers of that. So what we've been trying to do is to try and understand what is it that is happening there. And that's worldwide. This has been our major focus of research for the last 15 years or so. But what we discovered was that the whole function of this protein called TDP43 is actually to help balance stability of a very specific product of DNA, something called RNA. There's a whole bunch of proteins that conspire to that. And the way to think about that is that if I gave you a blueprint to build a house, everybody understands what a blueprint is. That's your genetic code, right?
Right.
You're standing around on the job site waiting for the truck to come by with all the lumber. And the genetic code has said that you need a mixture of 2x4s and 2x10s and some, the whole 9 yards of it there. And it arrives to you and you build a house. And that end of that house is the human being, right? So there's a middleman in that. That's the RNA. It brings the material out to where it's needed. It synthesizes it there. And then you've got the right stuff on site. Now imagine somebody was goofing off and filled the truck with the wrong. So you get a house full of 2x4s. It's going to take you a while to build something and it won't even look right. It probably doesn't work. that has to be regulated. That has to be taken care of. And so these proteins that are gathering up inside these cells that are degenerating are part of what regulates that RNA, that transport of material to make sure you get the right stuff to the right site. So this goes wrong in ALS. What we've discovered is another protein in the same family of regulatory, so it's part of that shipping the material out there. But for everything the one that is present in ALS does wrong, this other one does right. And so the two of them seem to balance each other off. And that's been the real breakthrough, is to discover a protein that actually, in fact, can counteract the effects of this toxic protein. Now, what's really unique about it is that even in the experimental models that we've done, and we look in the spinal cord of those experimental models, whether it be flies or whether they be the mouse models for it, the train wreck is still there. And yet the cell's perfectly fine and working its way through it. And that's a major understanding change for us in ALS, is that we thought these aggregates had to be gotten rid of. This might tell us maybe they don't need to be. And so it puts us on a new pathway of understanding that you don't actually have to get rid of those. You just have to restore the normal cellular balance.
You mentioned flies and mice. Tell us about how far you've gone with these experiments, and then maybe we'll talk about how far there still is to go.
Yes. So this has been almost 20 years of work, a little bit over 20 years of work.
Wow.
To get to here. And just by way of a quick understanding, the first 10 years before those 20 years, really spent my life trying to understand, did all those 2x4s look right? So everything that was delivered to the site, were they the right size? Were they the right shape? Could they be used? At the end of the day, it turned out that they were all fine. It's just that not enough of certain ones were brought to the table. And so you had shortages of it. And then you can imagine that leads to a mess of how the whole skeleton of the neuron is put together. And discovering that there was a regulation of all of that led us to this identification of this new protein, RGNEF. And we did a lot of work initially to show that it was the yin and the yang, so to speak, of the toxic protein. And that led us to say, all right, we need to start putting this into animal models and see whether it makes a difference. So the first one was the field has done a really good job on developing different kinds of models of ALS. None of them are actually ALS, but they are the basic processes that are at play. So one of them is done in flies. And so we can take the human bad protein, its gene, and put it into a fly. And if you do that, it's born with this gene turned on. And so the fly does not live more than 14 to 21 days at most with a motor paralysis. That's not, up until that time, it can't climb, it can't fly, right? Normally, it should be out to 60 to 80 days. So then, if we took the protein that we're interested in, and we know its gene, and we put it into the same fly at the same time as we put the human gene. So now it's born expressing not just the bad one, but the one that we think is good. Flies are normal, right? In fact, what you just said was exactly what we said,
Right?
Which was, this is, and it wasn't a matter of just a minor trivial change. They had normal lifespans. And when the other flies couldn't climb up the side of a wall of a test tube, they were quite happy doing so. And they had normal wing structures. And we can force these genes to go into the eye. And so the ones with the bad genes, the eye is destroyed. We put the good gene alongside of it, the eye's normal, right? And yet, when we look at the spinal cord of those flies or into the brain, we find that the train wreck is still there. But there's no reaction to it. Everything else is doing fine. So that was kind of a peel us off the ceiling moment, right? When we saw all of that, really excited about that. Which meant next step is to take it into mice. And taking it into mice means that you really can't have the gene in the mice turning on at the moment of birth. You need to have them to mature and become young adults. And we ended up putting in, and it's a model that's used worldwide, is to put in a genetic variant of the bad protein that forces the motor neurons to behave as if they had a motor neuron disease and give us largely the pathology that we see in the human cases. So when we allow the gene to be turned on as a young adult mouse, that mouse will die with a motor paralysis as early as 10 to 12 days, often in around the 3-week mark. And it's striking. There's just no mistaking that this is happening to it.
Is that equivalent to the span of ALS in a human being in their decay? Or are you doing it faster?
We're doing it lots faster. We're doing it lots faster because we're sending it into all the neurons simultaneously that are motor neurons. And so the whole 9 yards goes. Yeah, so it's much, much faster. Well, what we also found was that we didn't need to create a double transgenic construct. We didn't have to put our good gene at the same time. We were actually able to put that gene into a virus that is perfectly happy wandering around without causing any problems whatsoever. And it tracks to the central nervous system. So we make a minor modification to the virus and it goes right to the motor neurons. So the motor neurons that are in trouble because of the bad protein. And so we can show that this virus is being expressed in those cells, that the protein, either the full size of it, and think of it as a yardstick, it's a big protein, or the first 2 inches of it, which is a smaller, where it gets rid of a whole lot of other things. Both of them significantly change the disease course. So the first time we did it, we have a picture of Lucy up here. Lucy was our first mouse that we treated. Her sister with a genetic abnormality died at around 13, 14 days. Lucy was still alive around 18 weeks, right? Big change. And then we needed to do much larger numbers and such. So long story short, that's what our publication was 2 weeks ago, was to demonstrate that not only did we understand how this thing functioned, we presented the fly data, then we presented the data from the mouse. And then we also demonstrated how these two proteins actually interact with each other, right down to using artificial intelligence to show the two protein structures linking together. It's like a hand and glove kind of process, or a lock and key is another way to think about it. Think about the lock as being the bad protein, and a small fragment of the new protein just needs to lock into a specific site. So that's what we found.
And you said that your experiments with mice showed that you don't have to introduce these at the same time. So theoretically, and I guess we'll talk about what theoretically means in a moment, but theoretically, if it proves out, that would mean ALS would develop in somebody naturally, and then you would introduce whatever you create with this good protein to neutralize it, essentially.
That's going to have to be the scenario, because and although there's a lot of work being done on trying to understand how can we detect ALS before its presence, right? Is there a test that we can use? We're not there yet on that. So we are going to be at the mercies of the diagnosis once things are in play, and then introduce a treatment.
So what is the theoretically here? How do we get from this great results you're seeing in mice to, I guess the next step is a clinical trial in humans? What's the process here?
Yeah, it's a good question. So, a lot of people think of it as like a marathon of work as opposed to a bunch of sprints. I actually think of it like a hurdles race. We got quite a few of them ahead of us. So, our next steps in all of this are, even as I referred to earlier, we're talking about the 2 inches at the end of the yardstick, right? The first 2 inches of being important. That's still, when you think about it in terms of something being administered, gargantuan. It's huge. So we need to get it smaller. And we've designed those smaller fragments now that we, at least in the test tube, know that they will interact. They'll still be, that lock and key is in place. So we need to test that in the mice going forward, because we need to now show that the smaller fragment is biologically active. We need to demonstrate as part and parcel of that it's safe, and that we have markers beyond just the motor changes that would allow us to detect that this thing is being effective in real time. So there's a lot of work to be done there. But we're also going down 2 other paths. And that's why this is an international study that we lead doing this. So our colleagues in Italy, so Emanuele Beretti and his group in Trieste, have been working with us now for almost 8 years on this as well. And they've developed a cell culture model in which we have been able to demonstrate that you get the balance between these 2 proteins and you can disrupt them. And we know that there are somewhere around 3,500 to 4,000 drugs that are out there that are off-label, might have a molecular mimicry. So he's going to be looking to see, is there something already existent that's been through the channels and might work? If he finds one, then that will be sent to us here, and we'll do all the animal experiments. And then we have a colleague at UBC, Sriham Subramanian, and his group, where his expertise as a cancer biologist and biochemist is to design new drugs that wouldn't require a virus. And so his group is working on another pathway there. And then our group, led by Christian Droppelman and his team here, is doing the virus work. And so at the end of the day, I tell people we're swinging for the bleachers using 3 bats.
maybe so people who don't live in this world can get a sense of it. For you and your colleagues as people who treat ALS patients, who work with families, how big is this news in your world? What's some of the reaction been to the publication? Like, what's happened over the past 2 weeks?
It's been amazing. So first off, amongst my science colleagues, right, those of us, and in Canada, we're an incredibly closely knit community and really highly productive in ALS. People don't actually know that. We're amongst world leaders in this as a collective for it. Has been very much like ours, which is that we've been around long enough that we know that these things can go off the rails. Translating from a mouse to a human being has not proved to be what we had hoped it would be, say, 25 years ago when we first started using these kinds of models. So there's a lot of kind of, wow, this really changes our thinking. This is a breakthrough going forward, but we've got the hurdles ahead of us, and let's see where we land.
Nobody wants to get their hopes up too much yet.
That's right. The second has been, and it's been really gratifying from patients and from really right around the world that we've been hearing from people, families that I took care of, their loved ones, or have and still taking care of, other families sort of reaching out and saying, the most important thing about this is that it might not be there to help me, but we can at least see a horizon now that collectively you know what you're aiming for. And that's something we didn't have before. We all knew that we would get there, and it's been really important. The third piece of all of that is that, there's this sense of, where we use this fear of using the word cure in ALS, and very, very appropriately so. But I point out to people that, amongst ALS cases, somewhere between 8 and 10% will have an inherited form, right? So it's passed from generation to generation. And if we look at all of the genes that we now know that are involved in all of that, and there's now over 100, there's a small cluster of them that are very, very common, are the leading ones within that. We now know that 2 of those, although extremely rare, when treated with very specific genetic manipulation, right, in adulthood, we are actually seeing arrest of disease progression, and we're seeing improvement in motor function in some of those patients. And so if the response then to that is, yeah, but those are really rare, right? My response to that is, not if you're the one who's got it. And the second is, not if you're talking 5 years ago when we didn't even know that was possible. So, that has taken all of us as clinician scientists or as researchers working in this field and said, look, there's an endpoint here. And we now know that, will we be able to return function to everybody? No, I don't know that by any means whatsoever. But do I think we are actually on the right track now to, can we actually mitigate this disease? And that's actually the title of the paper, is mitigation.
The last thing I'll ask you then are the practical questions for people who are close to this disease. I know you cannot actually put a timetable on it, but when you say, it won't be here for me, but the important thing is it might be here for other people, what is a ballpark timeline for these kind of trials? And second, I guess, is when you look at how this treatment is created or how you hope to create this treatment, what are the chances that it will be widely available, affordable? Because I know new groundbreaking treatments for diseases like this can often not be that.
Yeah, terrific question again as well. So, one of the big things that has helped us here, so Jim and Louise Temerty and the Temerty Family Foundation have provided us not only the original funding to get past that fly right into the data, but then funding for the next 5 years to get this thing to the point where we're doing early clinical trials, what are called phase 1. And when we were talking about it almost a year ago to this date of doing it, they asked me the same question: what's a reasonable time window? And we think that window is 3 to 5 years to be at a phase 1 study. It could be faster if there are lookalike drugs. So if our Italian colleagues come up with things, or if our colleagues at UBC actually come up with a new design drug, which I wouldn't be surprised if they did, then that may be shorter. But I think a healthy window is 5, and that's if everything goes well, that there are no issues for that. The issue with regards to making sure that this is available to everybody, that to me is one of the paramount process behind all of this. So, best case scenario, this is something that's already sitting on a shelf and is generic and can be used. Then we've got no hesitation in moving forward. The trials get funded and away you go for it. there's no doubt that raising new biologicals is not for the faint of heart from a cost point of view and the cost recovery. But we're looking as hard as we can right now as we're designing what does this look like as we get to that part to make absolutely sure that this is something that can be readily available to everybody. That it's not an economic hardship to be treated for this when you get the disease, because the disease is already an incredibly expensive illness to have. It's been estimated $120,000 and $130,000 a year with this by the time all the support. So our goal, certainly my goal in all of this, is to let's get it out there.
Dr. Strong, thank you so much for this. It's really fascinating. And obviously, I'm not the first one to wish you the best of luck.
Thank you so much. Yeah, I really appreciate it. And it's just really been heartwarming, the sort of response to all of this. We've got work ahead of us. Let's hope the hurdles are small.
Dr. Michael Strong, professor of neurology at Western University, as well as the Arthur J. Hudson Chair in ALS Research. That was The Big Story. For more from us, you can head to thebigstorypodcast.ca. You can also, of course, send us your feedback via email, hello@thebigstorypodcast.ca, or by calling 416-935-5935 and leaving us a voicemail. The Big Story is available in all of your podcast players and on your smart speaker if you ask it to play The Big Story Podcast. Joe Fish is the lead producer of this show. Robin Simon is also a producer here. Chloe Kim is our research assistant. Stephanie Phillips is our showrunner. Sound design this week was handled by Christian Prohome. Mary Jubrin is our digital editor. Diana Kay is our manager of business development. And I'm Jordan Heath Rawlings, your host and executive producer. Have a wonderful weekend. We've got an In This Economy episode for you tomorrow and a blast from the past on Sunday. The Big Story will be back with a brand new, fresh episode on Monday. And we'll talk then.
My name is John Cullen, and I want to tell you a story. It's a story about a scandal, broken relationships, gossip, rumors, money, corporate rivalry, and curling. It's the story of Broomgate, how a single broom, yes, a broom, turned friends into foes and almost killed the 500-year-old sport of curling.
It was a year I'd like to forget.
Broomgate, available now.